Ladies and gentlemen, welcome to this first session of WBC 2018 on BVD. My name is David Graham and I'll be chairing this session along with my colleague Dr. Tajima. So it's my privilege and honour to introduce our keynote speaker for this morning, Professor Joe Brownlie. I'm sure many of you know Joe, but for those who don't, just briefly to introduce him to you, he qualified from Bristol. I'll not tell you the year. Went into veterinary practice before doing his PhD, followed by a postdoctorate studies in Australia. In 1977, he came back to the UK and started research on the pathogenesis of a range of veterinary viral infections and has contributed significantly to our understanding of a number of those diseases, including BVD, BIV, foot and mouth disease, blue tongue and canine infectious respiratory disease, and indeed has been involved and has discovered new viruses in those species and also been involved in vaccine development. He's considered to be an expert in new and emerging veterinary diseases and has consulted widely about their pathogenesis and control. And it is, as I say, my great honour to introduce him this morning on the topic of BVD to give his presentation entitled The Great Challenge of BVD Eradication. Can we do it together? Thank you, Joe.
Thank you, Chairman. And thank you, Matoshi. I'm sure that you were involved in inviting me. Not many people would have done, but thank you so much indeed. It's one hell of a time to start a lecture, I have to say. There's only one thing worse, is coming to listen to it at this time. I came to Japan a few years ago, many years ago, and there was a wonderful, wise, gracious, courteous professor of veterinary pathology, Professor Yamanouchi. And I was beguiled by his stories and his fine virology. And over dinner, he showed me the Japanese writing. And I thought it was just like an artist's painting. And the next morning, he brought this in for me. And this, I said, that is fantastic. It was on beautiful paper, and he'd written it overnight. And it says, Professor of Veterinary Pathology. He's such a gracious man, I do believe that what it does say. But there's a royal seal you can see at the bottom. And I was so impressed, and I said, what is the seal? And that seal there, he said, well, my dear colleague, he said, that is Joe. So whenever you see the red sign at the bottom of a picture, you know it's Joe. My lecture outline is I'm going to talk about the virus, how it causes diseases, how do we control it, and how do we eradicate it. So within this group of viruses, it's a flavivirus, and that tells you a great deal about it. It's an RNA virus, and it's very mutable. And throughout this lecture, you'll see just how mutable it is. Down here are the human viruses, yellow fever, hepatitis C. But you can see there's no human analog of pestiviruses. And over here are the veterinary ones, many of which are zoonotic. But here's our group of viruses, BVD, classical swine fever, and border disease. Just a little bit about the molecular biology, because it helps to explain both the pathogenesis and the diagnostic, as well as our greater understanding of the genome. And here you can see it's a small virus. It's about 10 kb, and it has about 10 to 12 genes. That's all it has, mainly structural here, non-structurals there. That is the area, this 5' UTR, which allows us to separate the two groups. And I'll be talking about that in a moment. These ones here, these structural proteins, are the major areas where we develop our tests for antibody. And that one there is the area where we know which biotype we come. So I explain all those, but that's where on the genome these major genes are being used to characterize the virus. Just to set the scene, we know we've got two biotypes of this virus. One is non-cytopathic, so it grows in cells but doesn't kill it. and the other one is cytopathic, which will kill the cells. And we just thought in the early days that that was something that just happened in cell culture. But now we know it has a fascinating role in the pathogenesis of the disease. And this disease that we're going to be describing, eventually mucosal disease, is unlike anything else we've described in virology yet. And the way that this virus here arises is usually, or if not always, by mutation from the non-cytopathogenic form of the virus. So keep that in mind. There's two biotypes of BVD. We know that it will affect a lot of these major species, cattle, pigs, and sheep. And there is clear evidence, that it will affect other non-bovine species. And Caitlin Evans will be giving a talk, BV10 session, about non-bovine species. We also know that BVD, which is considered mild for a long period of time, can have a very severe syndrome. And this is what we get. We get dead calves, aborted calves. We get quite hemorrhagic signs within the animal. This one here is particularly in the intestine. And here we've got a virus that's come through, actually was within a virus, and these cows have all died from this virus. Now, this virus is actually a second type. It's a group 2. And we'll be hearing from Dr. President. These were the original papers in the late 1980s. And from that, and we're very lucky we've got Julia Redpath in the front row, terrifies me really, but she's very lovely, was actually one of the first people to understand that these were two different groups of BVD. So we've talked about biotypes, we're now talking about different groups, and this was a group 2. And what we find with these group 2 is, in the early days, was they caused severe disease, often fatal. Disease clearance, the virus clearance took a long time. Normally it's 7 to 10 days. This took very much longer. It had profound thrombocytopenia, so it's hemorrhagic, diarrhea, hemorrhagic disease, but it only weakly cross-reacted with the group 1 BVD. So now we quite clearly have two different groups of the virus. And I'm going to go on to show you that we've even got more than that at the moment. And how the diagnostic was in the 5' UTR, so they could be distinguished. And this gives you some idea. Here, what we've got here is this is the group 1 that we're talking about. This is the group 2. And now we have group 3 as well. And we even have, this is border disease, this is classical swine fever. So they're related, but sometimes distantly. And here's one that was an interesting story in Australia. If I can show you the next slide. No, I won't. There, you can see that Peter Kirkland, one of our colleagues, discovered this new virus that was killing pigs. And he's talking about tens of thousands of pigs. Couldn't find what the virus was. Eventually, he came back to find that it was one of our pestiviruses. So the point I'm trying to make here is that this is a very variable bunch of viruses, and we continue to go on. And we've got some nice papers here by Andre here on Hobie-like viruses. That's later this morning. And again, we've got Yoshima, you're going to be talking about Japanese BVD strains. It'd be fascinating to see what you've got. So let me just summarize that because I just want to move on. So we've got different groups of the viruses, group 1 and group 2. In each of those, we can have subgroups. So we can have group 1A, 1B, 1C, and onwards. And there's really quite a number of these subgroups, as we have with group 2. Beyond that, we actually have non-cytopathic and cytopathic biotypes in each of these groups. And then beyond that, we have viruses that are either high or low virulence. So this makes this quite complicated. People get very confused between biotypes and groups and subgroups. And that's how it lays out. This is some work that originally done by Anne Lindberg in Sweden, but then again by Richard Booth, one of my students. And this is actually analyzing some part of the genome of isolates from cattle. Each one of these dots is a cow. And what I want to illustrate, I appreciate you don't need to know the detail of that, but here's one example here. And this is the same farm, got the same virus. So it's very easy to be able to actually genotype them. Each farm seems to have its own virus. So on this, there's 5 cows, and they've all got the same virus. Very interesting. Here we've got the same farm with different viruses. So that's understandable. The viruses come in twice from various sources, and we've got different viruses on the same farm. What's really, really interesting is this one here. The top one here, this farm here, we've done this number of cattle, and it's different farms here, but with the same virus. And how do you explain that? And what has been interesting is 2 of these farms, one of them, they were not connected in any way, but they won't both send their cattle to an agricultural show. And it's quite clear when we've done the pathogenesis or the epidemiology that one of those cows was actually a PI and transferred it at the agricultural show to the other one. And another one we had, there was 2 cattle with the same virus in different farms. One was in the south of England, the other one was in the north of Scotland. And the only way that they'd had any connection was in embryo transfer. So the take-home message of this really is to say that we have the ability to actually show where these viruses are. We can track these viruses. And it might even be that they could be used in legal proceedings. We know where these viruses are. And so I think that's fascinating. So how much BVD virus is in a persistently infected animal? We'll come on to persistently infected animals in a moment, but I think you'll all know. So if we say it's a 9-month-old weighs 250 kilograms, with an infection rate of about 10 to 6 infectious viruses per tissue, then we've got 250,000 million infectious viruses in this animal. So then the next question is, that's a lot of virus. How much virus do you need to infect an animal? And the answer is, from our work and other people's work, depending on the strain of virus, between 10 and 1,000 viruses will infect an animal. You can see how many one of these animals could infect. It can infect a whole nation. So these persistently infected animals are highly infected. Cattle have 22,000 genes. BVD has about 10. And what is quite amazing is that this virus here has the ability to actually kill this animal. It is just mind-boggling that something that's so small, with so little genetic information, can have such a wide effect on these cattle. We don't actually really know how this happens, but what we do know is with the effect of this virus in cells, what normally happens in an infected animal, this one here is to show an animal acutely infected for the first time, never seen the virus. And when we infect it at day 0, what happens by about day 3 is we start to get a viremia here that usually is cleared about day 10. Nasal shedding, again about day 3 we start, and might be up to about 2 weeks. And after that we get a slow immune response that goes on and rises to a plateau at about 4 to 6 weeks. There's lots of information in there, but I just want you to take home the message that this is a short-term infection that is cleared quite quickly. What we do know, and we've done some work here, is that although we think it's cleared as far as isolating the virus from nasal shedding or from viremia, the virus does last a lot longer. But I haven't got time to go into that. So what do we get with this acute period of time? We get fever and leukopenia and viremia and nasal shedding. We think the virus is cleared, and then we get an antibody response. That's typically what we expect to get. Can I read? What does it say? Keep going? Oh, I keep going, right. The non-cytopathic virus does damage. Now, what sort of damage does it do? Although it's non-cytopathic, it actually does more damage than the cytopathic virus. Reproductive, neurological, agalactia, I think we know all that. But let's just deal with immunosuppression, because that's really the basis of many of the diseases that we deal with. Now this is we've done some work and others have done some work. This is looking, transcriptomics, looking at the effect that the virus has on cells. We were very lucky to have some cells from Julia, cell line of leukemia cell line, BL3 cell line, and we are able to put in our viruses and see what happens. And what is upregulated, you can see along here, a whole series of the one. The one I really just want to pick out, because it's so complicated here, is really the immune response. And what we're seeing here is that they're not upregulated, but these clusters, the major one is immune response genes are downregulated. We also get downregulation of some development genes, which might have an impact on the fetal development. But it's really the immune response that I want to deal with. Now, this is a horrendous diagram, but I'll take it through. What we're trying to do here is to show what happens when you have high and low virulence viruses. We wanted to see, could we see anything in the immune response genes? The yellow is low virulence. So these are low virulence viruses onto these BL3 lines. And this is upregulation on this side and downregulation on that side. So you can see a number are downregulated here. This is both, these are genes that are affected in both high and low virulence, but the pink ones here, the ones I want you to look at, this is you only see in the high virulence ones. And the ones that I really want to point out here, which have been so interesting, is that the greatest effect we get with high virulence is actually in here, and it's on the immune response gene. So the greatest effect we have is with high virulent viruses affecting these genes here and downregulating the genes. So we know that there's a difference between high and low virulence. We're not sure the mechanism yet, but we know things are going on here. We know that if we have dual infections with BVD and other microbes, and this is all experimental work that's been published over the years, RSV, IBR, Mannheimia, if you put BVD in with it, you get a longer shedding of virus, a higher titer of virus, and more disease. Again, in enteric infections, rota, corona, salmonella. What I think is particularly interesting is some of the papers we've got. And here Julia is talking at a symposium later this afternoon, a BVD-0 symposium. She'll be talking about the effect of disease that BVD has. Outbreaks of acute BVD, especially, you'll be talking, I believe, later this morning. And the one that I really would pick out is Dick Sibley's paper on BVD and bovine tuberculosis. A fascinating story of how BVD has profoundly affected a TB infection. So don't miss that. That's what he told me to tell you anyway. This is an interesting, when we eradicated BVD from the Orkneys, what happened having eradicated BVD, what the farmers found, they had less barren cows, less abortions, more and bigger cattle. In fact, the farmers complained that the sheds were no longer big enough to winter house their stock because they had so many surviving. You know, farmers are never happy people. So how does it cause disease? Well, the bull is really central to this. And one interesting experiment, it's only ever been done once, but it stands as really quite significant. Work done by David Patent. Here is a bull that was infected, naive bull, and this line here, there were many lines, is actually the abnormal sperm that he counted after infection, just acute infection. 7 days after, there were 10 to 15 abnormal sperm percent. 30 days, 35, 50 days up here, 90% of the sperm were abnormal, and even out to 80 days, over half the sperm were abnormal. But you might not have noticed that because this animal was pretty healthy, but it has a really impact on sperm quality and health. And we've done work here, this is work that's done over two experiments over the years. If you get BVD infection in early pregnancy in the first 1 to 24 days, you reduce conception rate from about 80 down to 22 in this experiment, and from about 80 to 33%. Now, you might not pick up BVD for that, you just see infertility, and you could put it down to a lot of different causes, but BVD has this potential. So this is what I would call the classic BVD pathogenesis. BVD will infect an animal in early pregnancy. The cow goes red because she's viremic, and the virus goes across to the fetus, and that goes red. And so both cow and calf are infected. The dam can make an immune response. So we've seen with acute infections, they can make an immune response. But the fetus, if it's in early fetal development, is not competent to make it and remains viremic. And in fact, when the point of tolerance comes, this virus becomes tolerized by the animal by central deletion. And so this animal never has the ability to get rid of the virus. And it goes right through its gestational period, and it's born, and that's a persistently viremic animal. Those are the PI animals that we've just been talking about. And they are central to the whole, this whole lecture and the whole BVD story. What we do with control of BVD is get rid of these. These are the major, major reservoirs. And if we just complete the picture, we now know that some of these animals will go on and they'll die. And the fascinating thing is that if they get superinfection with this second biotype, the cytopathic that we talked about, then they will die. And experimentally, we've done this work. And within 2 to 3 weeks, they die of this fatal mucosal disease. And that's because the cytopathic virus has piggybacked on the tolerance to cause damage. An experiment I've never reported before, but we've done, is that if you have a CP animal that doesn't have the CP virus, it doesn't die of mucosal disease. And some of these animals can live for years without having that, because it's a random event, the mutation to the second biotype. What we have if we have the mucosal disease plus the cytopathic virus is just what I've shown you, that they die of mucosal disease. The question then is, what happens if you give the cytopathic virus to an animal that's not persistently infected? What happens then? And in fact, what happens is that it makes an immune response and doesn't die. Now, the interesting question is, what's the role of that non-cytopathic virus? And what we've done in notobiotic calves on a number of occasions is that if we totally immunosuppress these animals with cortisone, and then we give the cytopathic virus, would that, in a way, allow this virus here to cause mucosal disease? And the answer is yes. And in fact, the role of the first virus is to allow the second virus not to be recognized and to go on and cause disease. Normally, the cytopathic virus here, where are we? This one here, would be recognized and cause an immune response. And if we use that second biotype, that cytopathic virus, to infect cattle in early pregnancy, what we find is that there's no fetuses, no PIs are developed, and we don't get any neonatal calves that are infected. There's a whole explanation that I don't have time to go into, but we know it's all to do with the immune response. This is cloned home Heinrich Voge's fascinating work with Cumulus. And here's a bull that he had in his, they were growing through in the stud there. And on 4 to 7 months, they tested the animal for BVD, as they test all their animals. It was negative. 11 months, they started putting it into the program to collect semen. And they found they had BVD here. And right through until they killed it at 22 months, it had BVD in the serum. So he came, and he came to see me in a meeting we had in Scandinavia. And being a guru, he said, "Can you explain that?" And I said, "Yes, certainly. The problem is that you missed it here, that was really a PI. That was your mistake." And he said, "No, it wasn't." I said, "Yes, it was." He said, "Well, I'm going to send you some tissue." So he sent me a lot of tissue. And what happened was, we know that Cumulus became infected here. So this is why it's red. But in fact, what happened, if I can just go back one, at this point here, what we find is that although it's excreting BVD in its semen, it's got a very good antibody response going on here. And so what happens now is that we've got Cumulus, which has got an immune response, but it's got infection there. And we know that because we looked, and some of our work here, is that we looked at the seminiferous tubules of Cumulus, and we've got virus here. Some of them, the seminiferous tubules weren't. And so we've got a privileged site for BVD here. So this is a really curious condition, Cumulus, and it's been reproduced. It's not a very common procedure, but it is there, and it has been reproduced. So a summary of this was the PI animal is the main and almost only reservoir. High levels of persisting virus and shedding virus. It causes considerable loss. Acute infected animals will be virulent for 2 to 14 days and produce animals in 3 to 4 weeks. And post-infection immunity is protective. So now we understand quite a bit about the pathogenesis. What I want to do a little bit now is move on to the control programs. We know what we've got to do. And the options that we have is do nothing, vaccinate, or just control the PI calves. And there's some really nice papers later this morning by Mike Kirby on vaccination for reproduction, and Andre from Portugal, who I went to see and had a wonderful time, looking at the vaccine interval. So those are good papers to go and look at. Or we can actually control PI and vaccinate. Now, what my dear friend Julia said many years ago was that, talking about America here, what can 40 years of vaccination, 160 currently licensed vaccines, there might not be that many now, but there were at the time, do to eradicate BVD? But when we're talking about BVD, apparently not much. It's still thriving. And although applied almost worldwide, just unsystematically vaccinating cattle doesn't even influence the high incidence of BVD infection. What she's saying, and I hope I'm right in saying, she's not saying that vaccines aren't useful. In fact, they're extremely useful, used correctly. But actually control and eradicate disease, you've really got to go and look for these fantastic super shedders, these PI animals. And so taking that into control, what is quite interesting is some of the European programs, we'll talk about in just a second, have actually not even used vaccination. But when you've got a very high level of BVD, you do need to use vaccines, but use them carefully. So the question is, how do we find these PI animals? You've got a PI animal here. This is a PI, this is a naive, this is a code, this is an immune animal. So in any normal herd, you will have a PI animal. You have some that are immune and some that are naive. And you've got to go and find this red one. How do you do it? Well, you can test for virus in blood, or milk, or tissues, very commonly test and tag, and I think we're going to hear quite a lot about that, and I won't go into it. From Linda, from the Netherlands, BVD quick scan, very effective, and also from Stefan on antigen detection, and then from PI survey in Japan from Mio. So those are nice papers, and I won't need to go into those. There's also an economic consideration. If you're going to look for antigen for a PI animal, you can go and look for virus immediately. And with test and tag, which is one of the tests, you can, if you've got 10 animals, you can test all of them to find this one here. And so that would cost you 10 tests to find that animal. The problem arises is if you've got 100 animals, I do 100 because then I can work out percentage easily, and you don't know any of this. You don't know where your PI animal, and because we know that the PI animal is normally 1% of the population, we'll say we don't know what this one here is, but we don't know where it is. So you've got some choices here. You could be either negative or positive at that point there. Come on. And so if I can go back, you've got to test 100 animals to find that one. And that's quite expensive. So you're faced with this one here. If you do have that one animal, what also happens is that because it's a persistent and super shedder, very quickly it will infect other animals, which will make an immune response. And that will go on and go on. And so you'll get a majority of these animals that are actually immune. In fact, the acutely infected animals, before they make an immune response, will also be responsible for spreading the virus. If you take a row of any 10, as we just showed earlier, it would be very unlikely that any 10, or even 5, would not have antibody. So by doing that test, you only have to test 5 or 10, you would know roughly whether you've got a PI animal in there. And so that's the basis of young stock screening. So taking 10 there, if you took any row of 10, you'd say, yes, there must be a PI animal in there. If you took that same 10 and there's no PI animal, they'd be antibody negative. So that's the basis of these tests. And there's an economic consideration of how you go forward. The disadvantage of young stock screening is you can't do it till they're 9 months old because of maternal antibody. The advantage of tag and test is that you do them immediately. So, can we find the PI animal? Yes, we can. Can we control BVD on farm? Yes, we can. But can we eradicate the virus? And I looked up what eradication was. I rather liked that. To eradicate something is to get rid of it, to destroy it, to kiss it goodbye. Isn't that wonderful? Eradicate, it comes from the Latin word meaning to root out. So when you yank out a weed by its roots, it's been eradicated. It's not coming back. So that's what we're trying to do with BVD. And several of the countries have done a wonderful job. But actually, the proper definition, I've looked this up, is that actually extinction is when the infectious agent no longer exists in nature or in a laboratory. So have we got rid of all the smallpox viruses in the world? That would be extinction. Eradication means it's permanent reduction to zero worldwide. So we rather loosely use this word eradication. Elimination is what we are really doing, is a defined geographical area, and control is at the local acceptable level. So this was at the Berlin conference in 97, Atlanta in 82. But I think we'll go on using eradication because we're used to it. But the strict term in the country term is actually elimination. So these are the countries that have done fantastic jobs here. These have got, the blue have got regional programs, the white have got national programs. Scandinavia led the world, and they've got rid of BVD. Other countries have done extremely well, Switzerland and Austria, and the programs here you can see in Netherlands and Germany, Berlin, and also in Scotland and Ireland. And if you look here, this is over a period of about a year and a half. You can see Norway have got, these are PI animals, very quickly. Once they made progress and had a national scheme, they did a very good job. And so did Switzerland. So good national program once you start to get rid of all your PI animals. That's all you need to do. Different EU countries have developed their own but different programs to achieve total elimination. And they're all based on detecting and eliminating PI reservoirs. And we're going to have, from our chairman, David Graham, we're going to hear what happens in Ireland. They've done a fantastic job. And also from Volker Moenig in the special session later on this afternoon, Control of BVD across Europe. So those will be well worth listening to. Let me just rather quickly, I'm still doing well. The most important reasons, this is in Norwegian, for a favourable animal health situation. I love that, a favourable animal health situation. Wouldn't we all like that? But the Norwegians behave very well, as you'll see. What they found, that they had to maintain a government veterinary service. We'd all like to do that. They had a legal framework from the very beginning. They had limited or restricted imports of animals and good organisation of the dairy industry. That's what they found was very important. Also, they have this wonderful word called dugnag, which means that they all collaborate, they work together. And if we're going to do something, we all do it. Whereas in the UK, I know of farmers who are delighted that the next farmer's got BVD because they'll be doing better than them. But what they don't realize is that it's a real risk to them. But so we don't have this word dugnag in our language. If only we did. So there's cooperation between authorities and farmers, and there's a science-based competence. In other words, they understand the importance of science and the importance of understanding how tests can be used and their validity. So successful disease control. What we need, the common theme through all the programs was a national strategic and leadership. Leadership is hugely important, and we're talking here particularly veterinary leadership. Strong educational programs to make sure that everybody understands what they're doing. A legal framework to make sure that we all do it. It's much better that we all do it. We can't eradicate an infectious disease unless we all do it. We can't have some people still having it. So eradication is a very big jump from control. Use your common technology for identifying PI animals. And it'd be interesting to hear from David Graham about the technology they've used in Ireland. It was the same, it was a technology that they all adopted, if I'm not mistaken. And so David would tell us about that. Central surveillance and monitoring, and adequate funding and resources. All those you need to have if you're going to take on an eradication program. And what I would say is, whoever wants it, owns it. And if no one owns it, don't start. And that's really important. And finding out who owns the disease is crucial if you're going to take this program. Is it government? Is it the industry? Is it both of them? Who actually wants this done? And then you can start your program. And what you also find out is that I talk about carrot and sticks, and I realize that some younger people don't realize that this is actually how you get a donkey to move. You can either put a carrot in front of it and the donkey will be so encouraged it will move forward, or if it won't, use a stick behind it to give it a tap to move it on. And you can use that in persuading people to do things. And so what would be the carrot for BVD? Well, free testing and compensation for PI animals. That would be a carrot. What would be the stick would be the legal framework. And what I would say is that there has to be a very big carrot to make everyone want to do it. In fact, it has to be a colossal carrot. And even then, they might not all want to do it. But you only need a little stick. Even the threat of a little stick. I can remember being beaten at school, and how many good points I got at school never compensated for that little stick that I got beaten with. And so I never did what I did, which is having a bit of a thump. So that legal framework is all important, and I don't think you can get a national program without having that in place. So this is a big contract, my last slide almost. Agriculture is a national responsibility. European progress towards the control of infectious disease. So in Europe, there's a lot of progress and a lot of pressure to go forward to eradicate BVD. BVD control is a paradigm for a new culture of pharma cooperation, getting us all to work together. This DUGNAG, it's fantastic. We could all work together to do something. BVD, we can do it. We have only one reservoir. We don't have an insect vector. We don't have a wildlife vector of any importance. It's just those PI animals. And people have eradicated BVD, just finding those. It's a wonderful experiment for veterinarians to take responsibility. It's got a complex pathogenesis. Once you get your head around it, you're in charge. You can really make progress. And you can win this. It's not as difficult as Johne's or TB, which have lots of other complications. It's got a very clear diagnostic test for it. We've got one vector, and we can do it. And we should do it. Industry would provide infrastructural support. You hope that would be the case. Veterinary leadership is really important. As I look around the world, I find that becoming more and more of a problem, is that we're not getting our leadership in place. We're not having enough influence. And we really mustn't let that go. It's such a big problem. Some of the chief veterinary officers are, I wouldn't like to say emasculated because that implies male, but you know, they have a problem on their hands. Long-term commitment. You don't get eradicated disease in a 3-year program or a 5-year program. It's much more likely to be 10 or 15 years, but there is an advantage. And don't lose that commitment towards the end when you've nearly done it. Put more resources into it. It's an entirely new way of cooperating, that we all work together. That would be wonderful. And so, this is my last slide. Even in Proverbs, we know that we've got to look after them. And from me, thank you very much indeed.